Thank you very much, Luc, for your invitation. Thank you for the organizing committee for having me here today. And thank you for this great introduction. I'm not used to people praising the hypothalamus like that, so thank you so much. So I will share with you some of our last adventures in the hypothalamus, and more precisely, some neurons that are controlling reproduction. Thanks to them, we are here today. These guys control reproduction since the zebrafish to us. If these guys don't work properly, we won't be able to transmit our genes and perpetuate the species. And these neurons are quite peculiar because there are very few. There are only in the human brain 2,000, and they are distributed from the olfactory bulb to the hypothalamus, their cell bodies, so over a few centimeters. In mice, there are about 700 distributed, their cell bodies on 5 millimeters, but they all send long projections to a part of the hypothalamus that is called the median eminence of the hypothalamus, where they will release their neurohormone into the pituitary portal blood vessels. And this neurohormone, the GnRH, gonadoliberin, or gonadotrophin releasing hormone, will act on gonadotropins there to stimulate the release of ALS and FSH that will go into the general circulation to promote gonadal growth, gametogenesis, and also the release of gonadal steroids that will exert feedback action at the different levels of the axis. And these neurons are extraordinary because they, as I told you, they ensure the survival of our species. Why? Because they are not born in the brain. They are born in the nose, in fact, and they migrate from nose to brain, so from the zebrafish to human. Again, here you have a transparentized human fetus of 7 weeks of age. We have stained the peripheral nerves with peripherin, and here in green you see these GnRH neurons that are born in the olfactory paracord and start to migrate towards the brain, and here is a close-up. So here you see they're born in the nose, and then via the cribriform plate they penetrate the olfactory bulb, and then they will colonize the brain. And here you have the head of a mouse embryo showing also this migration of neurons during embryogenesis. And as you all know, so at birth it's interesting because the migration has ended, but we all know that we need a long period of maturation before reaching full maturation of these neurons, that is puberty onset in humans and all mammals. But this postnatal maturation can be divided in different phases, and actually every mammal 1 week after birth activates, these neurons get activated to induce what is called minipuberty. So minipuberty peaks at 1 to 3 months of age in humans and at 12 days postnatal life in rats and mice. And this will induce, this activation of GnRH neurons will induce the first surge of FSH that will talk to the gonads to promote steroidogenesis, and in particular the release of estrogens that will tell all the cells of the organism, including the cells in the brain, that a part of the development program has ended and we need to start another one. And this will help to mature the GnRH system, but also all the different brain areas, as you will see. And then after the full maturation of the system is at puberty. So here is we show the activation of the axis in babies and in both boys and girls. And we discovered a few years ago that at minipuberty, in fact, there was a fundamental switch that was operated in the control of the activity of the GnRH promoter. And this switch was flipped by the increased expression of microRNAs, including miR-155 and miR-200, that will repress repressors of the activity of the GnRH promoter, including CPEB and ZEB1. And if this switch does not operate correctly, then the neurons will lose eventually GnRH expression later in life. And intriguingly, so this we found that using animal models. And intriguingly, looking at the literature, we found that in fact miR155 was present on chromosome 21 in humans and chromosome 16 in mice. That has all of the genes of chromosome 21. And we wonder whether trisomy 21 or Down syndrome could be a pathophysiological application of altered minipuberty. So here you have in chromosome 16 or 21 in humans, you have the miR155, but also, in fact, you have 5 microRNAs that are present on this chromosome. And according to our previous study, 4 of them have been found to be enriched in the GnRH neurons, including miR155. So maybe the switch is altered in these people and mice. And here we are lucky to have several mouse models of Down syndrome, including the TS65DN, in which males, like in humans, show infertility. So only 1 out of 10 males are fertile, like in humans. And in females, they have subfertility. And so to start with, so we wanted to know whether the expression of GnRH at minipuberty was altered in this mouse model. So we crossed them with reporter mice that were expressing GFP under the control of the GnRH promoter, and killed the mice at postnatal day 12 at minipuberty, and then microdissected the area and performed some FACS sorting. And here we were very surprised to see that as soon as postnatal day 12, you have a decreased expression of GnRH in the Down syndrome mice when compared to wild-type littermates. And this is associated to an increase of ZEB1, one of the transcription factor that is a repressor of the promoter. CPEB weirdly is not changed. And you have a diminution of OTX2, that is a transcription factor this time that, in contrast to CPEB and ZEB1, activates the transcription of the promoter. And you see that is decreased. So they have indeed a marked change in gene expression, including the transcription factor controlled by microRNA. And then we wanted to know what was happening in adulthood on the expression of the microRNA, and we were expecting that the miR155 would be upregulated because you have 3 alleles rather than 2. And in contrast to what we were expecting, either the miR155 levels in the preoptic region, where are located most of the cell bodies of the GnRH neurons, was not changing, and the family of the miR200 that is not present on the chromosome 16 or 21, we saw that they were totally downregulated, and these are the ones repressing ZEB1, that is a potent repressor of GnRH neurons. So suggesting clearly that this increased number of microRNAs in Down syndrome could induce an imbalance in the expression of the microRNAs. And in adult mice, here it's not sorted, it's the total preoptic region. We see that indeed we have still decreased expression of GnRH transcripts, but also, as at P12 in GnRH neurons, increased ZEB1 repressor expression, and also CPEB, as you can see here, and we still have OTX2 that is repressed. So that was quite interesting. And then we wanted to know whether these changes were translating in the loss of the expression of GnRH somehow in these mice. So what we did is, first of all, we checked at birth, and here we saw that the number of GnRH neurons were identical in the 2 genotypes, so suggesting that migration was intact. But then, as you can see, we were starting to lose GnRH protein expression by immunoreactivity in adult age, while it was quite normal during the infancy and juvenile age. And then we wanted, because Down syndrome is known to be a neurodevelopmental disorder, and cognitive decline is exacerbated with age, and we wanted to know whether our change in GnRH expression could be somehow correlated or associated with change in either sensory perception or cognition using a simple test like object recognition test. And here we took P35 mice, juvenile mice, adolescent mice, if you will. And here we see that already at this age, the animals have impaired ability to recognize new odors in red when compared to the black ones. However, when we subject them to object recognition test, we see that both in males and females, these mice, Down syndrome mice, are as performant as their wild-type litter mates. However, so this is an age when you don't have impaired GnRH expression. However, in adulthood, when you have impaired GnRH protein expression, you see that they are still impaired with the ability to recognize new odors. But this time, we start to see cognitive impairment just when we lose this GnRH. So to link the two, so we were quite puzzled. So then, thanks to the transportization approach that has been developed by Paolo Giacobini in the lab, we were able to see for the first time the full contingent of GnRH neurons and their projections in the brain. And we realized something that we could not see when we were doing serial section of 20 microns, that there were actually a lot of fibers, GnRH fibers that, of course, were reaching the median eminence to control reproduction, but you had also a lot of neurites going towards the hippocampus or the cortex, suggesting that GnRH neurons could also control some the activity of these brain areas and why not cognition. But GnRH neurons, like many neurons, express GnRH, but also express glutamate, GABA, galanin, other things. So they could talk to these guys here, but maybe not via GnRH. And here, so thanks to the generation of another mouse model that was expressing GFP reporter in the cells that were expressing the GnRH receptor, we saw that actually, indeed, in the hippocampus and the cortex, you had many cells that were also expressing the GnRH receptor. And then to know whether these cells were important for cognition, had a function. So what we did here is we injected inhibitory dread virus bilaterally in the dorsal hippocampus ventral cortex here. And we tested the mice before and after the viral injection, doing the olfactory test and also object recognition test. So before, 1 month after the injection of the viruses, but this injecting saline. And then we waited again 1 month, and then we did activate this inhibitory dread, so that induces the hyperpolarization of the neurons. And here what is very interesting, so there is not a lot of mice, but it's quite convincing. So here you have the same mice, the 3 conditions. You see that they perform well in recognizing new odors and new objects to start with, even after viral injection when we inject saline. But when we inject CNO 10 minutes before the last task, we see that they are totally unable to recognize the new odor, and they are also unable to recognize the new object, suggesting that these cells are actually involved in some cognitive processes. So next, so we wanted to see what was using our transplantization approach again. So we wanted to look at the total brain. So this is a sagittal section, and this is a ventral section of a wild-type mouse, and here a Down syndrome littermate. And here you see pseudo-colored in green are the fibers controlling reproduction, let's say, and the purple and blue ones, the ones that are projecting to extra-hypothalamic areas, including the hippocampus, so we see them well in the wild types. But you see that in the Down syndrome mice, you still see the projections towards the region that will be involved in the control of fertility, but all the fibers outside the hypothalamus appear to lose their immunoreactivity for GnRH, and you can see here. So next, we wanted to know, because we saw that the MIR200 were downregulated in the preoptic region of the Down syndrome mice, we wanted to know whether we could, if we were rescuing the expression of this MIR200, whether it would do something to our mice. So to do that, we engineer a virus, either a scramble or a virus expressing MIR200B, one of the MIR200, that we injected specifically into the preoptic region, where are located the GnRH cell bodies. And here you see the viral injection, so it's very localized to the preoptic region. And here in green, in red, you see the GnRH neurons, and in green, the zone that was infected, and you see the GnRH neurons that have been infected by this virus. And this injection was very selective, because you can see here in the caudal region we see no green nowhere, but only in the median eminence of the hypothalamus where these GnRH neurons in red project to control fertility. And here we saw by qPCR that indeed we had overexpressed miR200 in this region. And doing that, we repeated, using dual RNA-scope analysis, we see this is a wild type, this is Down syndrome. We see less GnRH neurons in the Down syndrome and less OTX2 expression, that is the activator of the GnRH promoter. And when we inject miR200 into the hypothalamus, we see that we increase tremendously the number of OTX2 expressing cells, but also we awaken the GnRH neurons that had extinguished their transcript expression. And so we see the presence of much more GnRH neurons than in Down syndrome mice not treated with miR200. And here we see more expression of OTX2 in the GnRH neurons, suggesting that OTX2 could promote, for example, the expression of GnRH. And then here, interestingly, we tested the olfactory and cognitive abilities of our control mice. So when they were injected with scramble, we had no effect. And the same for the scramble injected Down syndrome mice. Here you see they are still cognitively impaired and unable to recognize odors. But then when we did that in the before and after the injection of the miR200, we saw that we were able to rescue the olfactory abilities as well as the object recognition abilities in our Down syndrome mice at levels quite comparable to the ones seen for the wild types. And thanks to a collaboration with Marie-Laurence Boutillier in Strasbourg, we have performed RNA sequencing both in the hypothalamus and the hippocampus of our mice, injected or not, Down syndrome mice or control, injected or not with miR200. And interestingly, we did not see much changes in gene expression in the hypothalamus, but we saw huge changes in the while the virus is injected selectively in the hypothalamus, we saw huge changes in the expression of genes into the hippocampus, and including 72 genes that were upregulated and 19 downregulated. And interestingly, the biological pathways that were affected were an axon enschisment, myelination, oligodendrocyte differentiation. And interestingly, among the 72 genes that were upregulated, 53 were normalized or downregulated when we injected the miR200 in the preoptic region, and these were normalized in the hippocampus. So it was quite intriguing. And to test whether our treatment was acting on myelination, we did also in vivo electrophysiology, looking at the speed of the conductance of a signal from the left to the right hippocampus. And here we see that this is impaired in Down syndrome mice when compared to wild types. And when we express miR200B in the hypothalamus, we increase the speed of transmission between the left and the right hypothalamus, as you can see here. So we have restored, maybe through remyelinization, the activity of this network. Next, we wanted to know what what was the actual reproductive axis, looking at the hormones in our Down syndrome mice. And we compared the results we had on Down syndrome mice with the results we obtained from volunteers with Down syndrome. And here we were quite surprised because we were not expecting that. So I told you that you have less GnRH expression, at least in adulthood. And so you expect that the axis is downregulated, but in fact at mini-puberty you see that you have increased LH levels, so it's more activated, and this stays also in adulthood when you have less GnRH. So more LH, circulating LH, more circulating FSH. However, this is not consistent, the testosterone levels were unchanged. But we were very reassured with what we were seeing here in mice when we saw the phenotype of the humans that is identical. That is to say, no testosterone changes, but more LH and more FSH, knowing that the Down syndrome are on the left and the control patients are on the right. So we have the exact same phenotype in mice and in humans. So we wonder, how could the reproductive system act like that? So we thought maybe there is an alteration in the rhythm of the release of GnRH. So to assess that, what we did is we collected blood every 10 minutes from the tail of mice and measure LH That is the surrogate marker in the general circulation of GnRH. And we saw that there was, this is a beautiful profile of GnRH secretion, 1 pulse every 2 hours, as you can see here. And here we saw that in Down syndrome mice, they still were able to pulse, but you see that the baseline is increased and that the amplitude of the pulse is truncated by about half, as you can see here, suggesting that the pattern of GnRH release was altered. So we thought that maybe, of course, doing gene therapy with MIR200 in Down syndrome patients is a bit difficult. So we went for the simple thing. Maybe we thought, what if we give back GnRH at different type of rhythm in our mice, whether we would rescue the phenotype. But first, we wanted to see what continuous infusion of GnRH would do to wild-type mice. And here we were very shocked when we saw that, is that if you implant an osmotic bilirubin pump delivering constantly the same amount of GnRH during 2 weeks, you see that within 2 weeks, the wild-type mice lose totally the ability to recognize new odors and recognize new objects. And this rang a bell because it's known, for example, that patients that have a prostate cancer or patients who have endometriosis, who take GnRH agonist that suppresses the GnRH axis, they develop cognitive decline and they have increased risk to develop Alzheimer's disease. So here people were not knowing why, and here we explain why. And here, indeed, when you do a constant infusion of GnRH, you see that you suppress totally the GnRH axis. It's flat. Then we thought, so we need to, if we want to rescue the pulsatile release of GnRH, we gain access to a miniaturized pump engineered by the Japanese that is programmable and in which you can impose the wild-type pattern to the system. So here you have a Down syndrome mouse without pump. You see truncated high basal LH. And here when we put a pump, the organism in fact takes the rhythm of the pump. So low baseline and beautiful peaks. And so in this Down syndrome mice, when we injected continuous infusion of GnRH, we did not further impair the olfaction or cognition. However, when we put on their back these pumps that were delivering pulsatile GnRH, we were able, as we saw for the MIR200, within only 2 weeks to rescue their ability to recognize new odor and rescue the ability to recognize new objects. So because these GnRH pumps, in fact, are used in humans since 40 years, because it's used, because you have some patients in which the GnRH neurons are never activated or do not migrate from nose to brain. It's called hypogonotropic hypogonadism and also the Kalman syndrome. And because these patients, when they come to they go to the clinic when they want to have a child, so these persons never have puberty and have no gametogenesis. So they go to the clinic, and then you can put on them a GnRH pump on the arm. And then after a few weeks, the woman will ovulate and be able to carry a pregnancy, and men will produce spermatozoids. So because it's a very safe treatment, because you use the natural drug at the natural rhythm, we could translate our mouse studies directly to humans. So but first we had to do a pilot study. So we included 7 patients to whom we injected 1 pulse of GnRH, 17 nanogram per kilo, every 2 hours. And here you see on the left the population before. So we were testing for cognitive test and hormonal test and also MRI before and after the treatment. And here you see clearly that the patients had very variable cognitive profiles. Some were performing almost as well as the mean score of the general population. This is the gray area, while some were quite cognitively impaired. But interestingly, all of them, after 6 months treatment with this pulsatile GnRH, they improve about 20% their cognitive abilities. So this, we need to take that into carefully, because it could be a placebo effect, because both the patients and the medical team were knowing that they were receiving a treatment, so the patients want to show they improve, and the medical team wants their patients to improve. But what convinced us that it was true, is what we saw using a functional MRI at the resting state, and looking at the default mode network in some sub networks that were known to be down-regulated in patients with Down syndrome. So here we found the exact same thing that was published in the literature. This pathway was down-regulated in our patient. And after 6 months treatment, we saw that the functional cognitivity of this network was multiplied by 5. So the neuroradiologists that analyzed the data were shocked, because they never saw such a change in such a short time in adult patients. So, voilà. So this tells us maybe that is true. So now we are conducting a new essay on 60 patients to see whether we can find again the same results. So here, I don't know if you noticed, but the alteration we saw in the GnRH system apparently occurs as soon as mini-puberty. That is to say, 1 week after birth on. So we wanted to know whether, if on the opposite, when you have exacerbated mini-puberty, if you have exacerbated mini-puberty, whether you could have also cognitive defects. So what would be the phenotype? So here, the CPEP, that is the repressor of GnRH, is controlled by nitric oxide. So the activity is stimulated by nitric oxide. So we looked in mice that were knocked out for nitric oxide, and to see what was happening. And here you see in brown that the mice knock out for nitric oxide synthase, neuronal nitric oxide synthase, sorry, have an exacerbated expression of GnRH in the brain, and they have also at postnatal day 12 at the moment of mini-puberty, and increased FSH levels, as you can see here, and they have hard time to come back to baseline at weaning. So we have this type of curve in mutants when compared to the black one in controls. And this rings a bell, because exacerbated mini-puberty is the characteristics of children that are born prematurely. Indeed, little girls that are born prematurely have a mini-puberty that is exacerbated by 300 times sometimes. And we all know that children that are born prematurely are at higher risk to develop neurodevelopmental diseases with mental and non-mental comorbidities. So we wanted to know whether the NOS could be found as a mutation in humans. So my PhD student who performed these studies at the time went to Lausanne to find, in a cohort of patients with hypogonotopic hypogonadism, to see whether she could find mutation in the NOS gene. And indeed, she found 5 different mutations, in very rare mutations in this cohort. And what was intriguing is that in some patients, it was associated with sensory loss, like bilateral hearing loss, or also intellectual disability, and also alteration of smell. So here, next, because of this phenotype of these patients, we next tested the cognitive performances in these mice, and we saw that they were indeed, in adulthood at least, had an impaired ability to recognize new objects, and also they had a problem to recognize new odor. Here it's not like the previous one. You see that they see that there is a new odor, because they spend 300% of their time there, but they don't integrate the information. And this has been shown in babies that were born prematurely. When you do visual tests, when you put a new object, the baby stays fixed on this and does not realize what is happening. So this altered minipuberty then could have maybe an impact on future development. So we wonder whether if we were able to rescue minipuberty by giving back nitric oxide during the minipuberty, whether we could rescue this phenotype. And why nitric oxide? Nitric oxide, in fact, is a drug, it's a gas that is given to babies that are born prematurely to save their life, to open their airways, etc. So it's already a treatment that is given to baby. So we gave the exact same dose that was given to babies to our mice in some mole incubators. And here you see that we were able to rescue minipuberty in these mice. And also we were able, so the treatment by nitric oxide or Viagra, the sildenafil that prevents the degradation of downstream signaling of nitric oxide, was able to rescue, just the treatment during this critical period was able to rescue cognition and olfaction throughout life. So this is a very intriguing finding. And this led to, contributed to the launch of a European project looking at the importance of minipuberty in babies born prematurely on their neurodevelopment. So at the other end of the spectrum, we were interested also by pathological aging, and we were intrigued by the fact that, for example, out of five patients with Alzheimer's disease, you have 3 women, and we wonder whether being a woman is a risk factor for developing Alzheimer's disease. And we put that in the context of a very beautiful old study published in the years 2000, where they looked at the rhythm of GnRH LH release in women that were young menopaused or aged menopause. So the young menopaused woman at 51-year-old, or the aged woman of 72 years old. And of course, the person at risk of developing Alzheimer's disease is not the 51-year-old one, but the 72-year-old one. So you must know that when you get menopause, then you don't have any more ovarian follicles growing, so you don't have a release of gonadal estrogens, and thus the brain is, and the GnRH neurons are constantly active for the next 30, 40 years. And because they are constantly active, maybe they are more prone to some insult and to get damaged with age. And indeed, somehow what is shown here, you see that there is a huge pulsatility in women during menopause, but then that this pulsatility decreases 20 years after, as you can see here, while there is no reason, there is no physiological reason, but just that suggesting that the GnRH neurons indeed get eventually exhausted, and you see the quantity of LH release is much lower in postmenopausal women. And to see whether this hypothesis could be true, so we looked at one of the most models for Alzheimer's disease or frontotemporal dementia, that is the Taito 22 mice from Luc Buet in Lille. And indeed, we saw in 1-year-old mice with this genotype that we had the same hormonal profile that we saw in the Down syndrome mice. That is to say that the amplitude of the LH pulses was affected in these mice, suggesting that what I just told you before could be true. And then to know whether, and here clearly these mice, it is known, have impaired ability to recognize new odor at the year of 1 year, and also are impaired abilities to recognize new objects. And here again, we put a pump on top of these mice. For 2 weeks, we delivered pulsatile GnRH at the rhythm, the wild type profile. And we saw that after 2 weeks, we were able to rescue olfaction and cognition in these mice, suggesting that the pulsatile GnRH is able to mobilize somehow the cognitive reserve of these mice. And then to finish, we were all subjected to the SARS-CoV-2 infection in the years 2020, and one of the questions was whether the virus was getting into the brain. And here what was intriguing to us is that we showed in mice, for example, that the GnRH neurons that have migrated in the brain, they still keep a contact with the olfactory epithelium via a dendrite, for example, or an axon. So the GnRH neurons could be infected by viruses that have their prime site of infection in the olfactory epithelium. And so we wondered whether the GnRH neurons could be infected in patients who died with COVID-19. And indeed, we collected 3 brains of people with COVID-19. And in one of them, we saw that, so we saw that GnRH neurons here in red that are bipolar, they do express the receptor of the SARS-CoV-2, CHRU, and we saw in one patient actual infection, the immunoreactivity for the S protein into this neuron, these neurons. And here in vitro, we were able to do primary cultures of olfactory plaquette of human embryos that will differentiate eventually in GnRH neurons. And we see that these cells can be infected in vitro by the SARS-CoV-2. And in all 3 patients, what was intriguing is we saw this weird shape of GnRH neurons. So usually they are bipolar like that. And here we saw in this patient, for example, disrupted morphology in GnRH neurons. And these GnRH neurons with disrupted morphology were expressing Cas-Pas3, suggesting that they were actually dying. So this we saw that in all the 3 patients. So, and interestingly, so what we did is we teamed up with some British colleagues to try to probe the health of the hypothalamic pituitary gonadal axis in patients with COVID-19. So here, what we do for that is we assess the levels of testosterone, for example, in males, and the levels of LH. So here in green, you have the patients that have normal hormonal levels. And then you have 2 categories of patients that are that have defects in the release of testosterone. As you can see here, they are below the threshold. And some of them are normal because testosterone has an inhibitory effect on the brain. And so when you have low testosterone, you should have activated GnRH neurons, so high LH release. So it's what happens in this person that appear to be normal. But here you have another category of people who have low testosterone as well as low LH, suggesting that the GnRH neurons are not functioning in these patients. And they develop thus hypogonadotropic hypogonadism. And there is a lot of examples of long COVID of patients that have the complaint to have low circulating testosterone levels and also low sexual desire and low mental abilities. So, and here, so interestingly, in UK, they received the patients 3 months after the SARS-CoV-2 infection and 1 year after the same patient 1 year after the SARS-CoV-2 infection. So in orange and red, you had the people that were producing low testosterone at the first visit. So the good news is that they all recovered normal HPG axis 1 year after the infection. But the bad news is that one person that was normal 3 months after, had a normal HPG axis at 3 months after infection, became hypogonadic. So suggesting that indeed SARS-CoV-2 infection could have very long-term effects. And we wonder whether, so here now we will study a new cohort of patients, 300 with short COVID, let's say, and 300 with long COVID that have been very well phenotyped, both men and women that have been well phenotyped for their mental and non-mental comorbidities. And we'll see whether there is an association between hypogonadotropic hypogonadism and these defects. So I would like to thank you for your attention, but also all the lab in Lille for their work, in particular these 2 brilliant PhD students, Valérie Leysen and postdoc Marianne Manfredi, who performed the studies on Down syndrome, and also my former stratospheric PhD student, Konstantin Shashlaki, who was at the initiative of the European project that we wrote together and we obtained on premature birth, nitric oxide, and postnatal development, neurodevelopmental disorders. And also my collaborators in the CHRU of Lausanne who performed the experiments, the study on Down syndrome patients, and Professor Wajid Dilo who performed in UK the experiment, the collection of data on people with COVID-19, and the funders for their help. Thank you very much for your attention.
Thank you very much, Vincent, for this exciting talk. So at least
I have a question. Did you look at 2 on amyloid deposits in hypothalamus in Down syndrome? And if yes, is the structure of 2 on amyloid is similar to that is observed in Alzheimer's disease or different?
Yeah, that's a really cool question. In fact, here the population of people we studied was between 18 and 30 years old, to be sure not to have Alzheimer's disease neuropathology. But it's true that Down syndrome patients at the age of 40, 100% of them start to develop pronounced neuropathology of Alzheimer's disease. And what was interesting, so this is the follow-up studies to see whether this treatment with pulsatile GnRH could help also cognition in older people with Down syndrome, but also maybe on people with Alzheimer's disease or with frontotemporal dementia to help mobilize their cognitive reserve. So this is interesting. And concerning so here, the physiopathology, we were not expecting that we would have an effect on it. However, there is a Chinese group that published the same year than us in 2022 a Nature paper showing that FSH that is increased after menopause could play a role in the pathophysiology of Alzheimer's disease, accelerating the deposit of amyloid beta and tau. And interestingly, the Down syndrome patients we had have elevated FSH levels, and when we treat them with GnRH, pulsatile GnRH, we lower the FSH level. So by lowering FSH levels, that could have a worsening effect on, or accelerating effect on the neuropathology. Maybe we can indeed help to slow down the neuropathology.
questions?
And I'm curious if there are any studies in patients that show any link between early hypogonadism and any type of dementia, like the risk of developing dementia.
Yeah, so this is a beautiful question that I will try. I submitted a grant in which I will try to address that using mouse and primate models that are more short-living than humans. But clearly, so also here what is intriguing is patients with, it's more and more known that patients with autism, for example, they have also an increased risk to develop Alzheimer's disease later on. So here there are some cohorts in France, like the Marianne cohort, that collect samples from birth in people at risk to develop autism. And here the plan is to see whether these children have exacerbated or not mini-puberty. So to try to connect and then use animal models or primate models to actually test this hypothesis that is wonderful. And is Alzheimer's disease a neurodevelopmental disorder? Yes, maybe.
Yeah, but in clinics it has been proved that accidents of life or chronic stress may maybe deregulate the axis between the synthesis of cortisol and the hypothalamus.
Yes.
And
exactly. So maybe, for example, the GnRH neurons, for example, in women after menopause are constantly active. and so they are more prone to react badly to insults. And some of these insults could be the stress indeed. For example, a woman who is highly stressed, maybe it will alter the function of the GnRH neurons faster, and then let the floor for the neuropathology to increase or accelerate. Or yes, this is possible.
Thank you very much. That was a fascinating talk. I was thinking back to the hypothalamic circuits. I think that there is some control of oxytocin of GnRH neurons. Have you looked into the patients if there is a link with oxytocin as well, or if that supplementation can?
That's another fantastic question. And actually, we are currently interested by the Prader-Willi syndrome. The Prader-Willi syndrome is another neurodevelopmental disorder in which the children develop intellectual deficits, less pronounced than the people with Down syndrome, but also morbid obesity. And one of the therapy for these children now is the treatment of oxytocin during early postnatal life. And indeed, it's known that the GnRH neurons do have oxytocin receptors, and oxytocin could control the activity of the GnRH neurons. So this is something that we are currently evaluating both in mouse models and we'll try in patients too. So thank you for the question.
Questions? I have a question. Do you did you ever look at a possible function of GnRH neurons in locomotion or in the motor system, which is also related to the same kind of neuroanatomical pathways?
Yeah, that's another cool question. So I don't know whether there are projections. We did not look. We should do a transplantization of the animal for the spinal cord too. But indeed, there is two possibilities for GnRH neurons to reach their target, either synaptic contact or dendritic release like the oxytocin neurons, but also volume transmission via the CSF. Because it's known that at each episode of GnRH release into the circulation, you have an episode of release. You found at the same time GnRH in the CSF. So GnRH could be also acting via volume transmission, and so it could reach the spinal cord via the CSF. That's highly possible.
I have one suggestion. You should probably look at Kennedy's disease, because they are mutated in androgen receptor, and there's so it's an expansion of polyQ expansion in the androgen receptor, and I'm not so sure it's been properly evaluated in terms of reproductive axis and
the name of the disease?
Kennedy's disease. It's a motor neuron disease. Okay. Spinal bulbar muscular atrophy, and there are good mouse models also. Wow. David.
Thank you, Vincent, for this wonderful talk. You know my question, because we keep talking about that with GnRH, and I was wondering, with the result you will have with the clinical trial, if the first data are not fantastic with peripheral administration, maybe we should consider having a stronger impact with either intranasal, because I think it could be administered intranasally, or brain infused, to be sure, because it looks like being invasive, but in fact it's not, and the Down syndrome is so dramatic. So I think it was trying a more adapt way of administration.
That's a wonderful idea indeed. And so for the nasal injection, I think it might be a bit delicate, because you need the pulsatile release. And so when we look at women that are under the pill, and when you look at the pulsatility of the GnRH release, and women under the pill have no cognitive defect, so the oscillation, it varies. The lowest pulsatility you can see is 1 pulse every 6 hours. So it means that we should do this nasal injections every 6 hours. So it means during night too. So this might be a bit difficult. Exactly. So, what you say about intracranial infusion of GnRH, this could be a very good solution because, as I told Luc, one pulse of GnRH in the blood is associated with one pulse in the CSF, and we think that what transports GnRH from the blood to the CSF are a specific type of cells we are studying that are called thanatocytes. And in Alzheimer's disease patients, these thanatocytes are degraded. So indeed, maybe in this population, the peripheral pump won't work, and thus we will need the intracerebral injection.